Hello, my name is John Sferrin. I'm on faculty here at University of Wisconsin-Madison. And my job today is really to tell you more about the CMT1A's progress on CMT1A and continuing on with the themes that Gilles just introduced. And I'll tell you from the outset, I won't be able to tell you everything about CMT1A in a half hour. But in the next session, we'll have some excellent presentations from three experts that can give you a little bit more of an in-depth snapshot. But I'm going to try to give you an overview, really of the depth and breadth of our efforts for CMT1A. So anyway, I'm really excited to be able to be with you today because certainly these virtual conferences have a very wide outreach. So I'm going to tell you a few things you hopefully may have heard already. But when we talk about CMT, this is affects nerves. And these are different pictures of nerve fibers. And there's a few terms that you need to know to understand some of the rest of the presentations here. So nerves occur in bundles. And the white part here in this picture, or the blue part in this picture here, are really the parts of the nerves that transmit nerve signals from the spinal cord to a muscle, for example. And the coating around it, which is this sort of white part here, or shown in black in this picture here, this is what we refer to as myelin. And this is formed by Schwann cells. And this myelin is not only important to protect the nerve fibers, but it actually makes nerve transmission much faster. And you heard from Dr. Shai that one of the ways that CMT is diagnosed is by measuring the speed of nerve conduction. So I like to think of myelin making nerve transmission from something like 3G to 4G. Some of you might be enjoying 5G right now as well. But myelin is really an important aspect of peripheral nerves, especially the long nerves that transmit to the various muscles in hands and in feet. So myelin proteins are known to cause this issue we refer to as demyelinating CMT, and this could be classified as CMT1 or sometimes CMT4. In contrast, problems with nerve fibers or the neurons themselves and the extensions of these, which are referred to as axons, that was what we referred to as axonal CMT or CMT2. So essentially, I'm going to talk mostly about CMT1A during this session here. And CMT1A was, first of all, the cause of this was, first of all, established in the early 90s. And what they found is that on the chromosome 17, that there was actually a duplication event on that chromosome. And this duplication event causes an extra copy of this gene known as peripheral myelin protein 22, or PMP22, to occur. And this actual duplication event turns out to be the most common cause of CMT, and it's classified as CMT1A. And in fact, this cause of CMT accounts for roughly half of those that have a genetic diagnosis of CMT. So in essence, the problem is that we have 3 copies of the gene rather than 2 copies of the gene. And every gene in your body produces a protein. And so the amount of protein is just simply too high in CMT1A. So instead of being right at the 100% here, it's 150%, so to speak. And one of the major strategies that's being pursued is simply to kind of reduce the output of the gene, just turning the dial down on each of the genes so that you can bring this level of PMP22 protein back in the normal range. So this is really simply what we're trying to do, is to dial that down. Now, we also know that part of what causes CMT1A is something called cellular stress. And probably the best example I can think of right now is all of you are aware of the fact that there's a cargo that's piling up in different ports of our country. And this accumulation of cargo is really causing stress to the system. because it's accumulating here and is not able to be shipped out to where it's supposed to be. Now, we also have a similar system in the cell. To make myelin, the PMP22 protein has to make its way out to the cell surface. And to do that, it has to go through a gateway, and this gateway is called the endoplasmic reticulum. And if you have excess protein, it begins to pile up in the port, and this accumulation of PMP22 in this port, we refer to as the endoplasmic reticulum, causes stress to the system. And there are several therapeutic strategies that are being tested to try to alleviate that stress. So essentially, we have too much of the protein, and it accumulates in a wrong place in the cell, and that's really the cause of CMT1A. So in essence, then, we've really tried to put together a comprehensive strategy for CMT1A, which really draws on 4 different areas that I want to outline here. I'm going to talk a little bit about genetic therapies for CMT. These are exciting new technologies that have recently attracted FDA approval for other disorders. And these come in different flavors like ASOs and gene therapies. And we've been very active on this front to try to speed development of this kind of therapy for CMT1A. We have ongoing testing of drugs for CMT1A as well. These would be just small molecules or drugs that could be used to either alleviate that stress I said, or just to reduce the levels of PMP22. Now, we are very active in really trying to promote company partnerships and identify those things that are required for companies to be able to invest into therapy development for CMT1A. And you'll hear a lot more about this in the next session with Mark Scheidler, who's been quite active in promoting different partnerships with companies. And one of the things that we've identified to be important for company partnerships is they need to know that there is clinical trial preparation for CMT1A. And there are a number of important things that need to be provided in order for companies to be able to do a successful clinical trial for CMT1A. And we're going to talk a little bit about biomarkers. But first of all, I just want to show this because this are several pieces of the puzzle that we really need to bring together in order for us to bring multiple ways of treating CMT1A to clinical trials. So first of all, we'll talk about genetic therapies for CMT, and these come under different flavors like ASOs and gene therapy. And really, the template for this is already established. There's a disease known as spinal muscular atrophy. This is a very devastating disease, which can infect infants and is really life-threatening in the most severe forms. And even though it's much more severe, it does affect the same motor neurons that are affected in CMT. And until recently, there was really no treatment for this very devastating disease. But in the last few years, there have been 2 new genetic therapies that have gone through clinical trials and have received FDA approval. And this includes AAV gene replacement therapy and also ASO therapies. And these are genetic therapies that have really been transformative for individuals with SMA. And these have shown dramatic effects, and they've been now approved by the FDA. And this really gives us a game plan that we can follow for CMT, because many of these strategies can also be applied to CMT, although we'll have to adapt them in certain ways, which we'll talk about in a minute. So anyway, this is an exciting area where some of these types of therapies are coming to clinic, and we definitely want to enhance this pathway to bring this to CMT1A. So I wanted to define this a little bit more. When we talk about a gene therapy comes in many different types. This can be the use of genes or genome editing as a treatment. And this usually involves introduction of some genetic material. It can be in the form of DNA or RNA. And if we can deliver it to the right tissues of an individual, we can use that to modify or replace a defective gene. And as applied to CMT1A, gene therapy approaches can be used to reduce the levels of PMP22. Another incarnation of genetic therapies is the recent technology of genome editing, or CRISPR-Cas9, which is a Nobel Prize winning technology from scientists at UCSF and many others in the field. And in this case, you can inactivate one of the 3 copies of PMP22. And there's actually a company that's working, trying to bring this into clinical trials as well. So this is an exciting area where really these new technologies are approaching the clinic for many kinds of disorders. And there is a pathway for this to be applied to CMT1A as well. So one of the early successes of this was the use of antisense oligonucleotides for CMT1A. And antisense oligonucleotides are just very small pieces of DNA. And these have been known to reduce levels of a specific gene, and the different mechanisms can apply here. And the CMTA established a collaboration with a company known as Ionis. And they were able to provide data that in 2 rodent models of CMT1A, that application of ASOs was enough to reverse the neuropathy, and even some improvement was seen in these rodent models of neuropathy. And in fact, this confirmed that reducing the levels of PMP22 is a viable approach for treating CMT1A. And this technology was very exciting from these lab studies. But at the same time, it also raised some challenges in that delivery of these ASOs into nerves, to the Schwann cells of nerves, proved to be somewhat challenging. So there's been a lot of work in the field by many to try to improve the delivery of ASOs so they can be effectively and efficiently and safely delivered to Schwann cells. Now, this initial collaboration was with a company known as Ionis, but in the meantime, there are other companies that have ASOs or related technology known as RNA interference that are definitely interested in CMT1A. And some of those companies have publicly disclosed their interest, and others are developing experiments to establish if they can come into this field. But we're very excited about this sort of technology as being applied to CMT1A. And we really have a proof of principle that it could work. The other thing that I really want to really emphasize is the fact that when we have any kind of genetic therapies, we really need to be able to deliver to the Schwann cells that exist in nerves here. So this picture here shows the brain and the spinal cord and the nerves that reach out to the feet, and nerves that go down the arms. These are the peripheral nerves that have the Schwann cells. And so we really need to be able to deliver anything that is therapeutic to these nerves in order to either replace a non-functional gene or to lower the levels of PMP22. But there are many approaches that are being tested right now. And if we can develop ways to deliver this effectively, this will benefit not only CMT1A, but also many other forms of CMT, including CMT1B and CMT1X and some types of CMT4. And in fact, this technology will be required to allow us to use things like CRISPR-Cas9 or genome editing for CMT1A. So again, this delivery problem is fundamental, but there are many approaches that show some promise, and we're hoping that this will enable clinical trials sometime in the near term. As another incarnation of this type of therapy, we have been adapting AAV gene therapy. So AAV is a virus that's actually a very safe virus that can be used to deliver therapeutic genes here. And one of the projects that's been funded by the CMTA is to fund the work of Dr. Cleopa, who's really a fantastic neurologist, but also a gene therapy expert, who's been working to deliver therapeutic genes to CMT. And then there's a collaboration with his laboratory and then my laboratory, as well as Stephen Gray's laboratory at UT Southwest. And together we're trying to test different vectors that will hopefully enhance the ability to enter Schwann cells. We are trying to incorporate this RNA interference idea to reduce the levels of PMP22. And then we're trying to make this system as safe as possible by restricting the effects to Schwann cells. And putting these three things together as part of this collaboration will hopefully bring a breakthrough to allow us to have better and more enhanced delivery to Schwann cells. So this is a very exciting collaboration that's also underway in parallel with the other approaches which I just mentioned. So I wanted to say just a little bit more about the drug development for CMT. Many of you are aware of the FARNext clinical trial, which has been in the phase 3 clinical trial. As it's been reported, they're repeating the phase 3 clinical trial to deliver to deal with a few formulation issues for their drug. But in addition to that, there are other some exciting possibilities that are really on the horizon. One is a company known as Inflectus, which has developed a drug candidate known as Cephin. This is an analog of an FDA-approved drug known as guanabens. And this targets this sort of a stress response, which I just told you about before. And there's some nice data to indicate that this will be successful in animal models of not only CMT1B and CMT1A. And they've already finished phase 1 safety trials in Europe, and they have phase 2 clinical trials that are in planning. These pictures here really kind of indicate what the issues can be. This is pictures from the ASO study, which I referred to earlier. You can see what normal myelin looks like. You have the nerves in white and the nice myelin around there. When you begin to lose some of those axons, you have less of them and they're much smaller. But with improved treatment, you actually have much healthier axons with much healthier myelin. And this is what we're looking for, these animal studies, the ability to restore myelin to really a normal state as well. In addition to this, CMTA has funded 3 different classes of drugs, which are under study right now in the laboratory of Dr. Laura Feltry and Dr. Rabbits at University of Buffalo, also in my laboratory here at the University of Wisconsin, and also one of my colleagues' laboratories, Luigi Puglielli, who has yet a third class of drugs that he's testing for CMT1A. Some of these drug candidates are actually FDA approved, and if they show promise in these preclinical studies, they could move to clinic relatively rapidly. Another class of drugs that has really shown some promise is a class of drugs known as HDAC6 inhibitors. These were first shown to work in another type of CMT, but within the last year, there's been a fairly nice study showing that in an animal model of CMT1A, that this actually shows promise as well. There are several companies that have candidate drugs for HDAC6 inhibitors. And so we're hoping that this will progress along and perhaps even come to clinical trials within a year or two or so. And then finally, as I mentioned, we've set up resources for drug testing to occur with pharmaceutical partners. And this is going to hopefully also bring new candidates that hopefully will also come to clinical trials as well. So again, we have a fairly comprehensive program for testing different drugs in parallel to the genetic therapies that I was just telling you about. So one of the important topics that has arisen as we try to figure out how we can bring therapies to trial is really looking at clinical trial preparation and biomarkers. And essentially, I chose to show this picture here. I was passing an apartment building, and there was a series of meters. I guess these are gas meters. And if one wanted to know how much gas is being used in the individual apartments of the building, you could look at each of these meters and see how well they're doing. In the same way, we need to have some kind of way of measuring neuropathy and seeing how things are going in individuals with CMT1A. And that way we can measure if there's any improvement or positive response to treatment. And again, there's been massive amounts of work gone into developing neuropathy scores that has involved Inherited Neuropathy Consortium. They've developed pediatric and infant assessments as well. But as Jill mentioned, CMT is a slowly progressive disease, and this creates some challenges for designing clinical trials. And in fact, if you just rely on neuropathy scores alone, a company really is looking at a clinical trial that might last 2 or 3 years, and you would need a lot of people enrolled, and so that just raises the cost and the investment that each of these companies would need to do a clinical trial. And so there's been a lot of interest in trying to develop biomarkers, which would be enabling for partner companies to be able to invest in development of therapies for CMT. And in fact, that's one of the first questions that companies ask us is, what does a clinical trial look like? Because they want to look at how that would happen in order to be able to invest in drug development or development of genetic therapies. So I'm going to tell you just a little bit about this. I just want to reinforce that the data that's been collected from all of these centers around the world, there's Dr. Shai's clinic at Iowa City, but many other clinics in the US, those in London and Italy, also in Australia. All of these centers have been working closely and interactively to do research on various things that are required for clinical trials, things like gathering natural history data and doing these sort of outcome assessments. And the CMTA, along with other funding agencies, including the NIH, have really made these sorts of studies possible. And the numbers of patients that have participated or enrolled in registries is just tremendous. So I think it's important to emphasize that this has really been a team approach across many centers around the world that are focused on CMT. So let me just give you a flavor of how some of those things work. So one of the newer things that the CMTA has invested in is sort of remote assessments, which are basically ways of using wearable sensors. And a project that's initiated by Katie Eichinger and David Herman at the University of Rochester uses sensors that can be placed on the torso and during the legs, and they can be used to measure things like sleep and posture, activity, and gait. Many of us have become used to the fact that our iPhone or our phone will track our activity or the amount of space that we've walked over time. And these state-of-the-art sensors are even more sensitive and allow very sophisticated measurements of these sorts of things. And ideally, these could be used in clinical trials. In addition, there are other neuropathy scores, there's functional outcome measures, there's health indices, all of which are tailored to try to identify the status of neuropathy. And hopefully these would be enabling for clinical trials as well. Another exciting development has been the use of MRI of calf muscle. And this actually came out of the muscular dystrophy field. And this is an MRI, and they've color-coded different parts of the calf muscle. And in muscular dystrophy, you see atrophy of muscles during the course of the disease. And the same thing happens within CMT, although at a somewhat slower rate. But nonetheless, this turns out to be one of the most sensitive ways to measure the progression of CMT1A. And we're hoping that use of this kind of assessment would allow clinical trials to proceed with a fewer number of patients, which lowers the cost and provide results within 6 to 12 months rather than 2 to 3 years. And this has really been spearheaded by Mary Riley and Jasper Morrow in London. In addition, we've been quite interested in trying to look at blood biomarkers, which involves measurement of proteins and RNA in blood samples. And we've been fortunate to collaborate with Sanofi, and we've published a couple papers in the last years in collaboration with Dr. Shai to look at different biomarkers. We have some that are released from axons. We have other protein biomarkers that come from Schwann cells. And we're also looking at RNAs that can be measured from blood biomarkers. Finally, another exciting new development has been the ability to look at skin biomarkers. And we've employed a novel technology known as digital nanostring analysis, which uses these sort of beads on a string here. This is an actual picture of these probes that can be used to detect RNAs in skin. So it turns out that there are small nerves that course through the skin. And from a skin biopsy, we can look at the Schwann cells that are attached to those nerves. And we can use that to measure the levels of PMB22. And we're hoping that this would be another way in which we could measure progress within a clinical trial. So essentially, all of these things are in development, again, to try to make clinical trial design more efficient. And this will really attract company investment into this. So as I envision a successful clinical trial for CMT1A, we think of that we would, first of all, restore Schwann cell myelin. This would lead to improved nerve function and nerve preservation. And then ultimately that really ends up impacting patients in a positive way to slow and arrest the clinical progression. But we need biomarkers all the way along this trajectory here, which would allow companies to have an early assessment of whether their treatment is having some success at treating CMT1A. So again, we believe all of these sort of outcomes are going to help us track successful treatments along this trajectory. So, so company partnerships, again, I'll not be able to cover them all. In fact, we've dedicated a session in the next period here for Mark Scheidler to kind of outline the efforts that have been made to establish company partnerships. This is a partial list of companies we've interacted with, and for many of them, we've tried to help them with drug testing as well. And we are excited about these efforts, and ultimately, we're hoping that all of these efforts together will bring us more shots on goal in terms of developing novel therapies for CMT1A. So with that, I just wanted to reemphasize the fact that we have really an all-star team assembled. We have certainly great neurologists that are among the best on the planet in terms of understanding CMT1A as a disease. We've been able to attract a number of different companies that are interested in applying new genetic therapies for CMT. And Dr. Kleopis, who will talk in the next session, will drill down even farther into development of genetic therapies for CMT. We're also, again, leveraging all of these different efforts and ultimately trying to provide the basis of successful clinical trials through many of the efforts that we spearheaded. So this has been my attempt to try to give you an overview of everything that's in play right now. And it's really too much to talk about in the time that I've had. But I'm really excited on many fronts of progress we've made so far. And I really want to emphasize that much of the success is based on the people in the CMT community who've been willing to participate in some of these studies, and important to show willingness to participate in clinical trials, which I hope will be coming in the near term. So with that, I think that's all I had to do. So let me
Thank you. There we go. Thanks, Dr. Savarin. That was excellent information. As someone living with 1A, I greatly appreciate these updates and everything that your team and everyone is doing to advance research for 1A and all of the subtypes. So thank you for sharing that information. We are going to end this particular session, everyone. And when it ends, again, you'll see at the top the session button. You can click on the session button and navigate to the next session. There are 3 being offered, one for type 1s, one for type 2s, and a session for type 4s. So please navigate to the one that makes sense for you, and we will see you in there. Thanks so much, Dr. Savarin.
Thank you.
Bye-bye.
Bye-bye.